So if anybody's here in person or watching the live stream, officially nothing has started. So you can ignore. Whoops, where'd it go? You can ignore the attendance words I'm going to give here in a second. I'll let you know when you need to write them down. I don't know where that person went. So anyway, if someone's watching the video later, so not today, not Morgan or Tasha or Kincaid, you don't have to write this down. But anybody who's watching the video, like I've said before, if you don't come to class and you have to watch a video because you missed it, that's fine. But you'll just have to sometimes jump through a few more hoops than everyone else does. It is an in-person class. So it's only fair that if you got to sleep in or whatever it is that you may have done to not be here live, then some extra hoops. So that being said, your extra hoops for watching the video. So again, not Kincaid, not Durgan, not Burton, not the rest of you. But for the rest of you, for those of you who are watching the video, let's say the attendance word is hoops. Like I said, sometimes you have to jump through some extra hoops if you're watching a video instead of joining us live. But anyway, just to make sure I'm not wasting anybody's time, you guys can ignore this if you want. Hopefully I'm preaching to the choir, but I'm going to give some announcements that I can put in record it with the 5 minutes we have left. Stuff I've already said, but seems to be having issues. Some people seem to be having issues with it. So for lab, again, you'll open that document. Actually, let me just show you. Why not? Why talk about it when I can show you? Let's look at how to do a lab in these 5 minutes. It's probably actually the only thing I can show you. Then we'll run out of time. Good morning. Oh, come on. Never mind. Now we've got 3 minutes and some change, so I won't do it. But yes, like I was saying, for the recording, for anybody watching the recording, when you open a lab, make yourself a copy. Like, let's talk about next week. You're going to open lab 3. You'll see it. There's directions on how to do everything I'm telling you. The directions are up top. Like, make yourself a copy by going to File and drop-down menu, make a copy, and make yourself a copy. And that's the one document you're going to work with all next week, right? You don't need to share anything separate with me, like here's the pre-lab, here's the lab. You'll open the document. You'll share it with me sometime before Sunday, right? Because that's when your pre-lab is due. Open that document, share it with me, and that's it. You don't have to share it with me again. It's shared with me. You don't have to make a separate document. That one document will suffice as your pre-lab and your lab. That's why it's all together. Another thing to say about labs is, what else? I'm trying to think of some common mistakes I've seen. Don't delete anything, right? So I know it might seem handy, but some people have deleted everything but the questions. But I don't need just the questions. I need everything, because I do try to change these labs in between every semester. So even though the lab you're doing is a lot like the lab they did last semester, it's a little bit different. So for that reason, I don't have all the questions memorized. So if you delete stuff, I might not know if all the questions are there. And also, sometimes somebody will write something down and I want to mark it incorrect. And then I'll remind myself, wait a minute, that's not what the introduction says. And I'll go back and I'll read what the actual lab says. And it turns out that you are correct and I give you the points. But it's harder for me to do that if you delete everything other than your questions. So again, with labs, make sure you don't. The only way you should edit labs is by adding stuff to it. Don't delete stuff. Don't rearrange stuff. Just add stuff to it. Any questions about labs? All right. Let's go back to attendance. Let's see. Kincaid is online. Abigail is here. I'm still going to try guessing since we still have a minute and some change. No, I don't think think so. Maybe. Mullins? Yes. Lewis? Okay, Lewis is here. Mullins is here. Oh, excuse me. Mullins is here in WVSU gear. Lewis is here. Cooper? No. Hunter was here. Right? Did I say Hunter was here? Yeah. Hunter's here. Melton is here. Both Meltons are here, I should say. Crowder's here in WVSU gear. Reinhardt? Is that WVSU shirt? I can't tell. I'm not wearing my glasses. Okay. So Reinhardt is here. Like I said, both Meltons are here. No Julia yet. Rogers? Nope. No. Got that one. Kennedy's here. Madeline is here. Hughes is here in WVSU gear. Galloway? Cook is here. Durgan is online. No. Burton is online. Kinder. Do we have? Yes, Kinder is here. Don't think. No Jones yet, correct? Oh, Jones is here. Sorry. Getting used to your face. Maybe Canterbury? Yes, Canterbury is here. Prowse? Prowse is not here yet. Hobson? Sorry. Ray? Fisher's here in WVSU gear. Let's go back to the top. Andrea. Cooper. Julia. Yep, is here. Rogers. Talbert. Kylie. Larissa's here in WVSU gear. Galloway is here. So far, other than the person walking in right now, everyone's been called, right? All right. Andrea? Hey, you got it. Okay. It's 8:01. So let's get started. The first word for attendance, unless you've been watching the video, in which case you have an extra word you have to do, but for everyone else, the first word for attendance is, let's just say acid, since that's what we're talking about. The first word for attendance is acid. And as I've been saying, just to remind you, if you guys are here watching this live, either in person or streaming live, you need to send me these words. What in the world? Send me these words before 9 a.m. So obviously for those of you here in person, those will be extra credit words. For those of you online, that'll be your actual attendance words. So the first word is attendance. This is where we left off. This is good. For those of you who had lab on Monday, this should be a review. So in a sense, you have the advantage right now because you've already been exposed to this. But for those of you who did not have lab on Monday, on this is good because now you're going to get introduced to this now. So then when we talk about it during lab, this will be a review. So here we go. Acids. The compound that releases hydrogen ions is what we consider an acid. You definitely need to know that for the exam. And for independent work, you can look this up. Let me tell you this. There are different definitions of acids. This is the definition that we use. Probably almost every Bio 101 or any version of Bio 101 around the country probably uses that definition because it's the most basic. So yeah, something is acidic if it has a lot of these H+ ions. The more acidic it is, the more of those it has. The less acidic it is, the less of those it has. So you should associate those H plus. So that's a hydrogen without the oxygen or without the electron, right? It's an H plus. You should associate that with acid. So something that accepts those hydrogen ions and removes it from the solution is called a base. But more importantly, I'm gonna circle this or put an arrow to it. As far as the exam is concerned, this is the bullet point that you need to know for base. A base is this. It's something that releases hydroxide ions. I just want to keep it nice and simple. And again, there's so many different ways of describing this. You can look it up for independent work. But as far as we're concerned, you should associate H plus ions with being acidic and OH minus ions with base. So keep that in the back of your mind when you're studying. Think of that. And there's so many, I mean, even already, even though I've just started teaching you guys this, there's different ways to describe the situation. So if I say something is very, if I say something is more acidic than something else, I'm saying it has more H plus ions than something else. Another way of saying the same thing is I'm saying it has less OH minus ions than something else. Does that make sense? Because in the exam, you need to think about that. Like there are questions on the study guide, which I use to write these exams, but I will reword them. So when on the study guide, it might describe it as something basic as having less H plus, or I might describe something basic as having more OH minus. So we're going to keep that in your mind, in the back of your mind, as you're studying acids and bases. So any questions about the definition of acid or base? All right, we'll talk about the pH scale. You guys are probably already slightly familiar with it. Let's talk about it. There's probably some stuff you might not know, or might, not probably, might. The pH scale describes acidity, which is the measure of the concentration of H plus in the solution, which you already know, right? So this last part of the bullet point, we already know from up here, right? The acidity is the measure of H plus ions in the solution. That's what the pH scale measures. Probably already knew that. What you might not have known, and I would assume not everyone here does, because every semester somebody gets this question wrong. But 0 is the most acidic, 14 is the most basic. You need to know that. 0 is the most acidic, 14 is the most basic. Another word for basic is alkaline. Another way to describe this important bullet point that you need to know, because again, you should think about this, different ways to describe the same thing. Another way of saying this is 0 is least basic. And 14 is least acidic. Make sense? I said the same thing. I just described it in a different way. Here's the other part that people usually miss. And this one I understand a little bit more. Like this one right here, the 0 to 14, to me that makes sense. I don't understand how many people, like why so many people miss it every semester. But this next one, I get it. It's a little bit more complicated. Each change, each unit for each step in the pH scale is a change of 10. A factor of 10. And I'll explain that on the next slide. So for now, let's ignore this bullet point because I'm going to describe it when I show you the next slide. So is there any questions about anything from there up? Any questions about this stuff? Okay, let's talk about this 10 times change thing. Well, we'll come back to that concept here in a second. Here's a visualization of the other stuff I said, right? So here's a basic solution. And the reason you can tell it's basic is, look, it has more OH minus ions than it has H plus ions. This one's acidic. How can you tell? It has more H plus ions than OH minus ions. And this is neutral because it has equal, right? It has the same number of OH minus ions as it does H plus ions. So any questions about that? Pretty simple, right? So simple, in fact, that you're probably not going to get a picture like that on the exam. That's like high school stuff. Here we go. Here's what I was talking about. Talking about the step of 10. So from here, we could say, if you go from 3 to 4, that is 10 times less acidic, right? Because you took one step. So that's 10 times less acidic. Or another way of saying that is that's 10 times more basic. Now, if you go from 3 to 5, so you go from, that's 10, times another 10, that's 100. So 5 is 100 times less acidic than 3. 5 is 100 times more basic than 3. 5 has 100 times fewer H plus ions than 3. 5 has 100 times more OH minus ions than 3. See what I'm getting at? There's so many ways to describe the same thing, and you need to know them. So again, when you're doing the study guide and you're studying this, think about all the different ways you could describe the same thing. But again, to focus on this mathematical part, from here to here, and I'm just doing that arbitrarily. Let me change it up, just to be arbitrary. From here to here is 10, right? So that's a 10. From here to here is 100. From here to here is 1,000. From here to here is 10,000. From here to here is 100,000, so on and so forth. Right? Every time you go up a step, it's a multiple of 10, which really says a lot. That means it's a big change in concentration. So that's what it is, right? This thing has a very, very high concentration of H plus ions. 14 up there has a very, very small concentration. But anyway, you definitely need to understand that. What you don't need to do is memorize all this, right? Those are just examples. There'll be no questions when I say, what's the pH of urine, right? No one needs to memorize the pH of urine. It's not important. But I will give you a question, or there is a question on the study guide where it says something like, compare, urine to grapefruit. And you could say, all right, urine is, let's see, so 10, 100, 1,000. Urine is 1,000 times less acidic than grapefruit juice. Or urine is 1,000 times more basic than grapefruit juice. Or urine is 1,000 times, more alkaline than grapefruit juice, right? All the different ways to describe it. The important thing here, well, I say important because I'm teaching to the exam right now. It's important because I see people miss this question all the time. Again, each step is a multiple of 10. Any questions about that? All right. This is good. I didn't make this. I just found it. I'll bring this up throughout the semester. I've already said it a few times. There's a lot of people who try to scare you with misinformation. And sometimes that misinformation is the truth presented in a scary way. So let's look at this. Does anybody know what dihydrogen monoxide is? Yeah, it's water. Not that I expect anybody to know that, but dihydrogen, the two hydrogens, monoxide, one oxygen, H2O. Yeah. Dihydrogen monoxide is water. That's another way of saying water. Most people aren't familiar with it. So dihydrogen monoxide is an acid with a pH level of 7. Well, to say something is an acid, in my opinion, is kind of a relative thing, right? So it's something more acidic than this, right? So, for example, you might say I'm tall because I think I'm the tallest one in the room. But if my my younger brother weren't here, you would no longer, you might not call me tall. You might call me short because he's taller than I am. So same thing with basic and acidic, right? And a lot of times you're comparing one thing to the other. So even though we know that water is neutral, and what you definitely need to know for the exam is that 7 is neutral, even though you know that, not everyone does. So far, this meme hasn't lied. Dihydrogen monoxide does indeed have a pH of 7. And one could argue that it is an acid, depending on what you compare it to. Then it says that's a higher pH level than any other acid. Well, that's a little bit of a stretch because, again, if you're going to consider water an acid, blah, 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 but it's not that much of a lie. The point is, here's something that's pretty much true that is presented in a way to scare you. Now, granted, whoever did this, I think this was facetious. No one probably actually put this out there as like really trying to scare you. But this is the kind of stuff people will try to do, whether on purpose or not. They're going to try to mislead you by either telling you the truth selectively or stretching the truth or whatever it may be, especially when it comes to chemistry. Like a lot of times I'll see something that says, if you can't pronounce the ingredient name, you shouldn't eat it. But that's so ridiculous because so many ingredient names of perfectly normal and natural chemicals, they have really weird names. But that's all very unimportant when it comes to the exam. I just wanted to let you know. Any questions about this? There shouldn't be because I'm not going to ask you any questions about it. Just an example. All right. What is the buffer? We already talked about this in lab. If you had it on Monday. But buffers are substances that minimize the changes in pH. So you should know that. Not verbatim. I probably won't ask you. I don't know. I might ask you the definition. But if I do, it might not be written just like that. So that's another thing I should point out that I don't know if I've mentioned before. When I say you should know the definition of something, a lot of times you don't necessarily want to memorize it verbatim. Like I don't care that you could spit some words out. I want you to know what the words mean. And this one is kind of simple, but there's other ones that are a bit more complicated. We'll cross that bridge when we get there. But yes, substances that minimize changes in pH, or substances that resist changes in pH, right? Same thing. Anyway, as you know, like I just said, they minimize changes in pH. How do they do that? Like I said in lab, for those of you who had it on Monday, they basically do the opposite of what's happening to them. So if you put in acid, right, which is a bunch of H plus ions, it accepts those H plus ions. When you put in base, or when things are basic, meaning there's not a lot of these OH ions, these H plus ions, then they donate them, right? So it keeps things, it resists changes in pH that way by doing basically the opposite of what's happening to it. So any questions about that? I'm gonna leave it at that. So I talked about the importance of buffers in lab on Monday. I'll talk about it again today in lab. So for now, I'll just leave it at that for buffers. Actually, I'll take that back. I'll just mention it really quickly. So the reason this is important is because that, if you have a huge change in pH, for example, in your body, it could kill you. And that would be a good independent work topic. I brought that up, I think, in lab, but just in case I didn't, look that up. Like I already told you, I think that your pH for your blood is about 7.4. And if it gets too high, you die. If it gets too low, you die. And I was just saying for independent work, you could look it up. What is too high? What is too low? It's not that much different than 7.4. It's a very narrow window. And you don't need to know it for the exam, which is why I'm not telling you and saying that you can look it up for independent work. Speaking of which, again, to remind you, you should have 10 points by now. And then by Sunday, you should have 20 points of independent work, right? Because this will be the end of week 2. Anyway, any questions about that? All right, in your old textbook, that would be the end of chapter 2, and this would be the beginning of chapter 3. But this textbook, your new one, combines them all together. So here we go. We're going to start what I call chapter 3, but what is actually, according to your textbook, 2.3. And this is the last portion before we have an exam. So keep an eye out for your emails and announcements, because soon I'm going to ask you when you want to take the exam. It'll probably be next week, and it'll probably be, your choice, like by majority. If almost everyone says we want to take it on Wednesday, if one person says they want to take it on Monday, 2 people say they want to take it on Friday, then most likely Wednesday it is. So if you have a preference, like I said, keep an eye out for that email. Reply to that email when I ask you what your preference is. And if you have a reason, let me know too. Because, I don't know, if you just say, I don't want to do it on Mondays because I don't like Mondays, that's fine. But if someone else then comes in and says, I really don't want to do it on a Wednesday because, I don't know, I have a vet appointment on Wednesday, then I'll take that into consideration. That's a little bit more important. Anyway, let's jump into it. Biological molecules. By the end of this section, you will be able to describe the ways in which carbon is critical to life. So you've heard the term carbon-based life form before, and now you'll understand why. You'll be able to explain the impacts of slight changes in amino acids in organisms. So everybody's heard of the word amino acids. You might not know what they mean yet, or what they are, but you will soon. And you should be able to describe the 4 major types of biological molecules while understanding the functions of the 4 major types of molecules. So not only be able to describe them, but also understand their functions. Any questions about the learning objectives? All right. As you already know from earlier in the semester, or earlier in the chapter, you need to know this for the exam. Cells are mostly water. Specifically, like you saw written, this is so hard to write on here, but it's 70, I really wish it wouldn't do that, to 95% water. Your cells are 70 to 95% water. That's a lot of water. However, the rest of most of the cell contains mostly carbon-based molecules. That's why, again, you always hear this term carbon-based life form. It's because most of the stuff, other than the water, is carbon-based molecules. Why carbon-based? Well, not that I'm going to ask you this. This is more like an introduction. So from here down, everything about this bullet point, you don't need to know, just an introduction. Carbon forms large, complex, and diverse molecules that are necessary for life. So those of you who've already done the drawing portion of Lab 2, this week's lab, you're already slightly familiar with this. And I don't remember if I mentioned this on Monday, so I'm mentioning it today for those of you who have lab today. When you draw the molecules, my suggestion is to draw your carbon skeleton first. Draw all the carbons, and then draw everything around that, because these are carbon-based molecules you're working with for the most part. Therefore, it's the carbon skeleton that pretty much stays the same. Everything else you're going to move around it, but for the most part, that carbon skeleton is going to stay the same. The word I'm not going to ask you, I'll put it next to this. These organic compounds are also called macromolecules, and they are carbon-based molecules. So again, mostly this slide is just an introduction. The only thing you really need to know from this slide is something I've already told you, which is that right there. Your cells are mostly water, specifically 70 to 95% water. So again, this slide is really just an introduction to all the stuff you're about to learn. Let's say the next word for attendance, or participation, excuse me. I'm just going to circle this thing, and you name it. You tell me what that is. I can think of at least 2 different ways to describe what's in this picture right here. So if you're in person or watching this live, send me the word of whatever you think that is. If you're watching the video later, take a screenshot of this thing that's circled and send that to me. Anyway, any questions about this introductory slide? All right, like I said, this is all about carbon. So let's discuss carbon really quickly. Carbon is a versatile molecule, as I mentioned earlier. That's not going to be a test question. That's also kind of an introduction. What you do need to know, and it's going to be hard for you not to know it after lab, is this bullet point. Carbon makes 4 bonds, right? Carbon makes 4 bonds. 4 single bonds, or 2 double bonds, or 1 triple bond in a single bond, whatever. It makes 4 bonds. And that is actually pretty relevant. As far as we're concerned, all the atoms and elements you learn about anyway, that's the highest number of bonds, which means it's the most versatile of all the ones that we talk about in this class. So because of that, that's why it's the main element. Because it has these 4 different bonds. It can bond all these different ways. They have all these different sizes and branching patterns. Not that I'm going to ask you that. This is all more like, well, what's it matter that carbon can bond 4 times? Well, this is just me explaining why that's important. Because we can have all these different shapes and sizes. And one of the things we're going to talk about a lot throughout the semester is this relationship between structure and function, or like the shape of something and what it does. So that is very relevant when we're talking about all this stuff, because we have all these varying sizes and branching patterns, right? All these different shapes. They do all these different things. These carbon molecules can do a lot of different things because they have a lot of different shapes. Here's some examples. Let's put an X in the middle here. These are just examples. You don't have to know it. But this is also from your old textbook. Carbon skeletons may vary in length, right? So there's a short one. That one only has 2. But it could have 3, it could have 4, it could have 5, it could have 500, it could have 5,000 carbons, right? It could just be a long chain of carbons, whatever. They vary in length. They also vary in their kind of bonds, right? So this one here, it has some single bonds, but it also has a double bond. What that doesn't show you, that would also give it a different shape, but your book, with your old textbook, really simplified things with that one. But we'll come back to that. Carbon skeletons may be unbranched or branched, right? So all of those are unbranched. They're just straight lines. This one is branched. So imagine this could keep going this way. This branch could keep going in this way and then have its own branch, right? Sometimes they'll ring up, right? So this carbon skeleton instead of ring. So many different examples. Again, the point here, not that you need to memorize any of these examples, is that carbon is very versatile, right? Because it can bond 4 times, it can make all kinds of different shapes and sizes. And again, it's this relationship between structure and function. So because it can make all these different shapes and sizes, it can do all these different functions. All right. You already know this, but we're bringing it up again. The big 4. Carbon can bond with other elements, and most commonly, at least when it comes to organic chemistry, which is all we're concerned about in this class, it bonds most commonly with hydrogen, oxygen, and nitrogen. Or again, like I said earlier, I think it was Monday, the big 4. The big 4 is carbon, hydrogen, oxygen, and nitrogen. So that's a review. And the little star, again, is just saying carbon most commonly bonds with these when we're talking about organic molecules. Any questions about this slide, this review of a slide? All right, here's an example. I put a big old X up top just to indicate, just to remind you this is an example. I'm not going to ask you any specific questions about methane, but it is a great example. It's you could say the most simple carbon compound because it only has that one carbon atom, right? The one carbon, and then it only has 4 hydrogens bonded to it. So really nothing fancy. One of the simplest organic compounds is abundant in natural gas, and it's produced by prokaryotes, which are basically like, as far as we're concerned, bacteria. So it's produced by bacteria in swamps. That's where we get swamp gas. It's also produced in the digestive tract of grazing animals such as cows. So, yeah, anyway, that's methane. Just an example. There's a lot more to methane than this. So if you want to look it up for independent work and look up some other facts about methane, you can if you want. Like I can tell you it's a greenhouse gas, but you could look that up. How potent is it as a greenhouse gas compared to carbon dioxide or water vapor, right? You can look all that up if you want. So any questions about this example? All right, here's different ways to draw it. Here's the structural formula. You do not need to know these. There's not going to be a test question where I show you one of these and say, which one of these is the space filling model? Which one of these is the ball and stick model? I'm not going to do that. I'm just showing you this because here's the way you need to know. Generally speaking, when you see any molecules drawn, not always, but usually when you see it in this class, it's going to be drawn with the structural formula. Whenever I ask you to draw a molecule, as you know, if you've been to the lab already, you're going to be drawing a structural formula. So this is the one you should be most familiar with. The only reason I bring these other ones up, well, because I teach out of the textbook, but also because I don't want you to get so confident with this. Like, all right, well, this is what methane looks like. And then for some reason you're talking to your buddies about methane. That's what people talk about, right? And then he pulls up this picture of a space filling model, says, that's not methane, this is methane. Because look, they look completely different. But now you know, right? There's just different ways of drawing molecules. Anyway, any questions about this slide? All right. This is another big old X, as in there'll be nothing specific about this on the slide. It'll be on the exam. This is kind of an introduction, some examples. So large organic compounds are the main molecules that are found in gasoline. They're also the main molecules found in the food you eat. Large organic compounds. And why is that? I'm about to tell you something important that we're really going to come back to later in this chapter and then later in the semester. The reason that these are used for food or for gasoline is because they're rich in energy. And I'll bring it back to something I said, what was that, last week? I don't remember exactly when I said it, but I was talking about the nucleus, right? The nucleus has protons and neutrons in it, which is something you need to know. So this is a good refresher. And I reminded you that protons are positively charged. And I reminded you that protons don't like being together, or positive things repel each other, just like negative things repel each other. So when you think about the nucleus, and I used uranium as an example because it had 92 protons. Think about 92 protons squished together, and they don't want to be together. And then what I said, and this is where it's relevant to this, what I said is basically however much energy it takes to keep something together, that's how much energy is going to be released when you break that thing. As far as we're concerned in this class, that's how you should think of it. And that's where we get this. These are large molecules, and it takes energy, right? It takes energy to build molecules. And generally speaking, the larger they are, the more energy it took, right? Because it'd be one thing if you think, that took some energy, right? But even without being chemists, you could look at this and compare it to this and think, all right, I bet this one took more energy than that one because it's bigger, right? You're putting more stuff together. It takes energy to put stuff together. Anyway, the point being is it took a lot of energy to build these molecules. Therefore, when you break those molecules down in chemical reactions, a lot of energy is released. And that's why that's why it's food. And that's why it's gasoline. Those things basically like batteries, right? These things are like batteries. And once you break them, they release that energy that they have stored. So that's why it's important. Another thing I want to point out, and this picture is not from your textbook, it's from your old textbook. This is also another great example of this idea of structure and function. So again, none of you are chemists, right? But you can look at this, and like, wow, this one thing, it's like those three things, right? It's very similar in their structure. And if you were to guess, and this isn't always true, but if you were to guess that their functions are similar, you would be correct in this case. Because of that long, straight shape it has, right? It has a lot of energy built into it. So does this. And its function is to store energy and then release it. So any questions about this slide? All right, here's another. This is what we were looking at earlier. Up there is the octane. So that's the gasoline molecule. That's what it looks like. These two things, there's two different ways of looking at it. So again, long molecule, right? If you break it, you release whatever energy is holding all those carbons together. And then this is part of the saturated fat we just looked at. Let me back it up. There's actually three of them. There's 1, 2, 3 of them all connected at the end. But yeah, so you can see, again, very similar. And again, none of you are chemists, so I don't expect you to look at this and understand its chemical properties. But again, there's this idea of structure and function. These things have very similar structures. They also have very similar functions. But again, I will put it next to this just to remind you. These are just examples. You definitely don't need to know this is a saturated fat, especially because we're about to talk about saturated fats, and you're going to realize that there's some exceptions to that thing. And you definitely don't need to know that that's octane up there. It's just an example. So any questions about that example? All right. So the unique properties of these molecules depend on the carbon skeleton, first of all, and also the atoms that are attached to that skeleton. I'm not necessarily going to ask that as a question. It's just something to keep in mind, right? So basically with this bullet point that I'm putting an X through, indicating that I'm not going to ask you in the exam, basically what it's saying is what I've been saying, just in a different way. That it's all about that shape, right? The structure and function, right? That's what I'm saying. The unique properties, i.e. the functions of these molecules, depends on their shape, right? Which is the carbon skeleton. It's the things attached to the skeleton. And some of the things attached to the skeletons are called functional groups. And I'm almost definitely not going to ask you what a functional group is, but I will use that term. So make sure you know what I mean when I talk about functional groups. Functional groups are groups of atoms that are directly involved in chemical reactions that are attached to the carbon skeleton. So a lot of times, not the examples I just gave you earlier, but a lot of times you'll have this molecule that's got the carbon skeleton, and really the whole structure function thing about it, a lot of times is the functional group. It's usually a lot of times it's the functional group that makes it do whatever it is that it does. And that'll make more sense when we start talking about these different types of molecules. Each one of these functional groups plays a different role in chemical reactions. And a lot of these biological molecules that we talk about in this course, or in this chapter even, they have 2 or more functional groups. I'm definitely not going to ask you that either. So these are good things to know, but I'm not directly going to ask you any of these things on the exam. Also, yeah, that's a good reminder. This is from your old textbook, right? So it's not this information's not even in your new textbook. Any questions about this before we talk a little bit more about functional groups? Okay, here's some examples. You definitely don't need to know this. This is from your old textbook. Even then, from the old textbook, I didn't require anybody to memorize any of this. This is some higher level stuff. But this is a great example. So you can see all these different structures and names of these different functional groups: hydroxyl groups, methyl groups, carbonyl groups, carboxyl groups, blah, blah, blah. Doesn't matter to you. You guys aren't biology majors. You are chemistry majors. But even here, you can look at these. And again, not being chemist, you could still look at these and like, well, those look different. They have different shapes. He just said different shapes means different functions. And that is true, right? So these different things with different shapes have different functions because of their shape. And then some other things, like we've already talked about, so this is a good reminder, polar versus nonpolar, right? So remember, a polar molecule is where the electrons aren't shared completely, or excuse me, are not shared evenly in that covalent bond. So a polar molecule has like a slightly positive side and a slightly negative side, and those things attract each other, right? So water is attracted to sugar. We'll talk about sugar later, but both of them are polar, so they're attracted to each other. Water and oil don't mix because water is polar and oil, which we'll talk about later, is nonpolar, so they don't mix. Long story short, not that you need to know this, but again, there's all these different properties. Polar and nonpolar don't mix, right? You don't want that. They won't come together. Polar and polar do mix. Nonpolar and nonpolar do mix. Excuse me, do mix. So again, these are just examples. The point here is these different shapes gives it, and also the atoms attached to it, give it different functions. Any questions about this slide? And again, I'll remind you, this is just an example. When you're studying, there's absolutely no reason to study this chart. On a molecular scale, many biological molecules are very large, gigantic, and they are called macromolecules. Definitely not going to ask you that. I'll just say this. Sometimes in this chapter, I'll use the word macromolecules. Sometimes I'll use biomolecules. And as far as we're concerned in this course, they're the same thing. Yeah. There's actually a fourth category, too, of biomolecules. So if you're writing stuff down, if you're a note taker, I wouldn't even write down these categories because there's going to be something else I'm going to teach you later. But yes, these are the three macromolecules. These are the three very large molecules. You are going to need to know about carbohydrates. You are going to need to know about proteins. You are going to need to know about nucleic acids. Those are all macromolecules. But then there's a fourth category of something called lipids. And technically, lipids are they're not macromolecules because they're not as big. But because of that, I don't like to get into the distinction between macromolecules and biomolecules. So again, I'm telling you, as far as we're concerned in this class, macromolecules, that word can be used the same as biological molecules. So again, just for your own knowledge, just to keep in the back of your mind, when we talk about lipids, which is the fourth category that we're going to talk about, it's not technically a macromolecule. That's just for you to know. You don't need to make that distinction for the exam. So any questions about this slide? All right. Macromolecules are polymers. Here's something you definitely need to know. You definitely need to know what a polymer is. You definitely need to know what a monomer is. And polymers are basically large molecules that are made up of monomers. You put a bunch of monomers together, you have a polymer. So to use some non-biological examples, a train, right? If you see a train hauling coal in All City or wherever, that train is the polymer. Each individual train car is the monomer, right? The monomers make up the polymer. Same thing with a pearl necklace, right? The whole pearl necklace is a polymer. Each individual pearl is a monomer. But yeah, so these molecules, other than lipids, because it's not technically a macromolecule, these polymers that we're going to look at are going to be long, these molecules, they're going to be long polymers that are made up of monomers. And for the exam, that's one of the ways you're going to distinguish between the two. So again, there's four categories of molecules we're going to talk about, and you're going to need to be able to distinguish between them for the exam, and that's one of the ways you're going to need to distinguish between them. So I say something like, which of the four molecules has, I don't know, polysaccharides as their polymer? And you don't need to know this yet, but you will. And you'll say, oh, yeah, polysaccharides, that would be carbohydrates, not lipids, not nucleic acids, not proteins, but the polymer for carbohydrates is polysaccharides. And then I might say, which of the four molecules has a monomer of, I don't know, amino acids? And I'll teach you this later. But then you'll know, all right, amino acids. So amino acids, that's the monomer that makes up a protein. Does that make sense? So remember, there's four categories of molecules that you're about to learn. And one of the ways you're going to distinguish them between each other is the name of their polymers versus the name of the monomers. So any questions so far about what a polymer and a monomer, what polymers and monomers are? All right, another thing you need to know is what brings them together, and it's called a dehydration reaction. As far as we're concerned in this class, every time we talk about a chemical reaction that brings things together in this class, I am oversimplifying a little bit, we call that a dehydration reaction. And really this should say, oh yeah, I could leave that. Links two monomers together. That's a little bit too specific, really, because technically you could have a polymer here and a polymer here. They're already polymers. And you could bring them together and now have one huge polymer, and that would be a dehydration reaction. So as far as I'm concerned, the best thing you should think about, or the best way to remember dehydration reaction is, let's just say, I'm going to cross through this and just say links molecules. Well, that's going to be really too hard to draw like this because it does that stupid line thing. I'm just going to abbreviate it like that. Links molecules together, brings things together. So again, whenever I'm talking about bringing things together, in a chemical reaction as opposed to breaking things, that's called a dehydration reaction. Now, just so you know, I'm not going to test you on this, but if you're wondering why is it called a dehydration reaction, it's because it removes a water molecule from the products, right? It removes a water molecule. That's why we call it dehydration. I'll give you an example on the next slide, but for now, are there any questions? Oh yeah, so this picture, this is a good example. These are actual polymers. This is a carbohydrate. I know they gave them little eyeballs, so that part's fake. But here we have a monomer of carbohydrates. That's a sugar. That's the monomer attached to another sugar, which is a monomer. Attached to another sugar, which is a monomer, so on and so forth. Right? You have all those monomers brought together to make a polymer, and they were brought together in dehydration reactions. So here we go. Here's a picture that's not from your textbook. Here we have a short polymer being attached to a monomer. This is just an example. We could have had a monomer on both sides. We could have had a short polymer on both sides. We could have had a long polymer on both sides, whatever. The point here, like I said earlier, is when we bring things together in this class, we just call that a dehydration reaction. And again, it produces water, right? It produces water. That's why it's called a dehydration reaction. Hold on, before I take questions. Yeah, let me look at it like this too. So if your chemical formula looks like this, I don't know, molecule A, whichever it may be, let's call that the short polymer, plus molecule B, whatever it may be, in this case, the monomer, gives you molecule C, which in this case is the longer polymer, plus water, that's the key there, plus water, H2O. That is a dehydration reaction. So there's a key, because you will get a test question on this. And that is one of the ways you can definitely recognize a dehydration reaction. If the water is on the product side of the equation, which will always be on the right in this course, not always in real life, but in this course, it'll be on the right. If you see water on the right, which is the product side, that's a dehydration reaction, right? Because you dehydrated these products, you dehydrated these reactants to produce water. So that's why it's called a dehydration reaction. Yes, question? No, good question. None of these are hydrogen bonded. The reason we know is because, and this will be important maybe later, remember a hydrogen bond is always going to be like this, like dash. So that oxygen is hydrogen bonded to that hydrogen. So when you see a dash, that's a hydrogen bond. Yeah, so but to your point, not necessarily to your point, but to address your question, it could be. None of these are hydrogen bonds. I can tell just by looking because they're drawn as covalent bonds. But that doesn't mean, again, these are just examples. So you could bring something together that had a hydrogen bond over here and a hydrogen bond over here. But when you bring them together, whatever you're dealing with, those are now covalent bonds, and that's what makes it a dehydration reaction. So it always produces a covalent bond, and it's only in this picture that those are covalent bonded to begin with. Any other questions? All right, we should move forward. So what's the opposite of dehydration reaction? Hydrolysis. Hydrolysis is the opposite of dehydration. That's what makes it a little bit difficult, right? If I were to just say you need to know what dehydration reactions are, that in itself isn't that difficult. But then I add this to it, hydrolysis. And the reason I was always confused with that as a student is because both names had to deal with water. Dehydration, hydrolysis, right? It always confused me. Not confused me, but made it harder for me to remember which was which as a student. But yes, hydrolysis is the opposite, right? So instead of making new molecules and bringing things together, we're breaking down molecules. We're breaking down bigger molecules to smaller ones. We're breaking these covalent bonds. So a hydrolysis reaction, again, breaks down things. And again, just so you know, to explain the name, the reason it's called hydrolysis is because it adds a water molecule, a molecule of water. So again, if I were to write this down, here we have molecule C plus water, using the same example I used on the last slide. If you get molecule C plus water, that's so ridiculous. I hate this computer so much. Anyway, that gives you molecule A plus B. So remember, when we were talking about dehydration reactions on the other slide, like I said, if the water is on the right-hand side of the equation, that's a dehydration reaction. So now I'm telling you again, because it's the opposite. If water is on the left-hand side of the equation, as you might be able to see through all those scribbles, it's hydrolysis. So hydrolysis means water is in the reactant side, and dehydration, water is on the product side. Any questions about this slide? All right. Let's see. We'll move forward. Here we go. Finally, the 4 categories. And there's no need to write these down if you don't want to, because we're going to have a long discussion about each of these 4. This is just the introduction. But the 4 categories of large biological molecules that are in every single thing that we know of that's alive: carbohydrates, lipids, proteins, and nucleic acids. I'm going to teach you about them. For the exam, you're going to need to know the difference between them. What are the polymers of carbohydrates? What are the monomers of carbohydrates? What are the polymers of proteins? What are the monomers of proteins? All those things that we just talked about, you're going to need to be able to differentiate between the 4. And like I said earlier, the first term I gave you was macromolecules, and technically lipids is not a macromolecule. And again, I just said that's just something for you to keep in the back of your mind. As far as we're concerned, we're talking about biological molecules. These are the 4 biological molecules we're gonna talk about. Does anybody know what this picture is right here? I mean, this one, I don't want to say it's a trick question, but it is a little bit trickier, because obviously you just want to say it's a bunch of trees, but it's a little bit more than that. And I was wondering if anybody knows a little bit. Yes. So this is their aspen. That's one tree. Technically, that's one organism, right? Yeah, it's a bunch of trees, but that is one organism that's propagated through roots. So the reason I included that is because, that's probably one of the larger organisms on Earth. And you can look that up for independent work. What is the largest organism on Earth? And you will get different answers depending on how you, what your criteria are for defining that. But yeah, that's one of the larger organisms on Earth. Does anybody know what this thing is? Yeah, it's a water bear. It is one of the smallest animals on Earth. And the only reason I chose those as examples, you definitely don't need to know anything about aspen or water bears, but it's a good example, because whether we're talking about one of the largest organisms on Earth or one of the smallest animals on Earth, they all have carbs, lipids, proteins, and nucleic acids. They're made up of the same stuff we are. But anyway, yes, you can look either one of those things up for independent work. What is the largest organism? What is the smallest organism? What is the largest plant? What is the smallest plant? What is the largest animal? What is the smallest animal? What is the, whatever you can think of that you're interested in. What's the largest bird? What's the smallest bird? Anyway, any questions about this introduction? All right, first thing we're talking about is carbs, carbohydrates. So, can anybody tell me where the carbs are in this picture? Pasta, very good. When I think of carbs, that's what I think of. Like if you ever go on a marathon, a lot of times the night before, people will do what's called carb loading, which is where they eat like a lot of pasta, for example, because you need energy, and you'll see how carbs give you energy. Yeah, so pasta, that's a good example. The other good example is this. It's Coca-Cola. And this is why I use this picture. It's because you need to forget what you know about, not forget what you know about carbs, but be prepared to add what you know about carbs. I think usually when people think about carbs, they think about your diet, which is a great picture for that, right? Because that's people eat and drink. That's your diet. Another thing people think about when they think about carbs is like breads and pastas, which is true, but there's more to it than that. So before we jump into it, just keep that in mind. I'm not asking you to forget everything you know. I'm going to ask you to add stuff to what you already know. Carbohydrates. They include sugars and polymers of sugars. And there's no need to write these examples down. This is just an introduction to what we're about to talk about. But the small sugar molecules found in here, that's carbohydrates. Those long starch molecules found in that spaghetti and the garlic bread that you can't see, it's off of the other part of the table, not in the picture, also carbohydrates. So again, this is just an introduction. There's nothing on that side that you need to know, except maybe this, but we're going to go into more details on this soon. They are polymers, right? So everything's a polymer or monomer that we're talking about, other than lipids. But now I'm also kind of hinting at the fact that the monomers are sugars. There's no need to write that down yet because I'm going to give a different name. This is just the common terms, right? Sugars is what we call them. But I'm going to give you scientific terms that you need to know here shortly. So any questions about this example slide? In animals, carbohydrates are the primary source of dietary energy, the raw material for other building molecules. I'm going to put a question mark next to that. Again, you're going to need to differentiate between the four categories. And that might be one of the ways I ask you to differentiate between carbs and other stuff. Carbs are the primary source of dietary energy, but obviously we know we get energy from other stuff, right? You can eat fat and get energy. Actually, I just showed you a picture of that earlier when I showed you a picture of octane and that hamburger and the french fries, right? That was a fat molecule. But yeah, the primary source is carbohydrates. And yes, that is also the building material. But if anything, know that for animals, carbohydrates are the primary source of dietary energy. So any questions about that portion? In plants, it is actually the building material for much of the plant body. So most of the plants are made up of carbohydrates, not like us. That might be our primary energy source, but we're not mostly made up of carbohydrates. Plants are. Plants are mostly made up of carbohydrates, and you need to know that. Whoops. Any questions about this slide? All right. The last word for attendance. I'm going to underline it. And I'm going to draw an arrow to it. And I'm going to circle it. That's the last word for attendance. And then I'll either see you on Friday or in office hours or in lab today. You guys have a good day.